or the finger bones and some teeth. And long back in the year 1970s and so on, the approach to date some artifact is radiocarbon dating. And if it is a human or any other animal fossil, then people will do anatomical analysis to fix whether it belongs to a human being or it belongs to some other organism. But in this case, okay. In this case, these two fossils look like it belongs to human being, but when they compare the anatomical structure, this finger bone doesn't exactly match with the modern human, and also the teeth doesn't exactly match with the modern human. So we all know, right, out of Africa model, like some lakh years before, from the Africa, the modern human being, we started migrating towards the north, and we occupy so many Europe and Asian locations, and then from there we slowly moved to Australia and everywhere. But the fossils that are collected in the Siberia doesn't belong to those who come out of Africa. So they are not definitely Homo sapiens, but the anatomical structure looks similar to the Homo sapiens. And at that time, we don't have facility to check those small bone structures. And then there is a very big limitation for radiocarbon dating. It can date only the fossils which belong to up to 50,000 years ago. If the fossil is way older than this years, we cannot deduct the timing from radiocarbon dating. And then the third one, anatomical analysis is nearly impossible because they didn't find any large bone structure. Everything is small bits and pieces. To solve this issue comes the field genomics. And like, how many of you are aware of the field genomics? You all know genetics, right? So genetics is analyzing genes and their function, and how each character, like my eye color, my hair color, and how I behave, inherit from parents to the offspring and then to the next generation. That kind of study is called genetics. And in genetics, we will study the function of a gene and how it inherits from one generation to another. Whereas genomics is the analysis of complete setup of a genetic information of an organism. If I take Homo sapien, then I have 3 billion base pairs of genetic information. Everything will be sequenced and computerized so that I can get information from them, whether I belong to a Homo sapien or whether I am a female animal, or those kind of information I can take from the complete genetic information of myself, say here. So that analysis is called genomics. And with the help of genomics, the issue with the Denisova cave was resolved. Let's see how. So from the small fragmented fossils, they isolated DNA, and then they study the complete genetic information, and they find out that it doesn't belong to Homo sapiens. Rather, it belongs to two other organisms which are similar to Homo sapiens. Say these two are cousins of us. Neanderthals are subspecies of Homo, and Denisovans are newly discovered human being which are closely related to us, and they are not Homo sapiens. So if we see Neanderthals, they lived some 40,000 years ago, from 4 lakh years, and they were first discovered in the year 1859. And the small bones and teeth that were extracted from the Denisova cave belongs to a subspecies of human called Denisovans. They lived from 2,85,000 years to 25,000 years ago. And they were discovered. Such species was discovered only in 2010, only after the arise of genomics field. Though from the earlier approach, we cannot conclude anything, right, from the bones. But after the genomics, we found out their period of existence, that they lived some 25,000 years ago, and then their mating behavior. Like the Denisova cave hosted Homo sapiens, Neanderthals, and as well as Denisovans. Inside the cave, they all made together, and there were hybrids of Neanderthals and Denisovans, and there were hybrids of Neanderthals and Homo sapiens. Though, like, they are like fertile or infertile, those kind of information we don't know, but we have evidence that all the three subspecies mated at some point of time. And spatiotemporal information, even for example, if you see this girl, she is named Denny, or the sample name Denisovan 11. She belonged to the Denisova cave, and her mother is a Neanderthal and father is a Denisovan. And even when we analyze the genomic information of this mother, it mentioned that she doesn't belong to the cave. She migrated from somewhere and entered the cave and started her life, and there she reproduced Denny. So such new informations can be obtained from genomic information. So once we became experts in genomics, every secrets of archaeology or every nook and corners of medical field becomes explored, and we came to realize a lot of information from those fields. So even like we know that this blue area indicates the presence of Neanderthals. They spread all over Europe, and the red indicates the presence of Denisovans. They spread all over the Asian countries. And even us, today, the modern human being, we shared at least 2% of Neanderthal DNA and at least 6% of Denisovan DNA. So for me, it feels like it's more interesting. Like, I didn't expect there is some species called Neanderthal, which is kind of we call colloquially caveman, and I have some part of his DNA in me. So these interesting facts were revealed once genomics were like start booming. So now let's enter into the lecture, what genomics is all about. Even before I mention, like, we get the genetic information of this species, that species, and we have this kind of information. How all these things are collected? May I know how many of you are from biology? Like, okay, thank you. So let's imagine you have pricked your hand or finger with some needle, and you have a drop of blood, and that drop of blood may contain millions of cells. And imagine you have taken only one cell, and inside the cell there is a nucleus which hosts the DNA, which is like a recipe book. Whatever you do, you can get the information from that recipe book. For example, if a dog chases you, then you have to decide whether I have to fight the dog or I have to run away from the dog. For that, there is a recipe based upon your previous experience. That page you can turn from the DNA, and if your experience is always for fighting, then immediately adrenaline hormones will be secreted, and you will take a stone and beat the dog. And if based on your previous experience, you are not that like brave to stone a dog, you will your brain will order you to run away from the dog. So DNA is exactly a recipe book. Whatever you need, you can turn that page and you can cook what you want and you can do. So, and human beings, we have say 23 pair of recipe books called chromosomes, and each chromosome can be elongated into a thread-like structure called DNA. And within the DNA molecule, there will be small fragments which are called genes, and from that gene we will have some protein that will execute the function. For example, like if I eat some sugar, immediately my body has to produce insulin, right? Then the recipe book again will be open, and there will be some pages where there is a gene called insulin that will be cooked up to produce insulin hormone. And now I will get the required amount of insulin to metabolize that sugar. So genetic information inside a cell is that way. There will be a long thread which contains a large number of genes. In human, there will be nearly 25,000 genes, and remaining are non-coding regions. They have some other functions. Here, we don't need all those information. So if we like navigate within the gene, it comprises of double-stranded helix, which has 4 nucleotides. Can you see there? Adenine, thymine, cytosine, guanine. So these 4 nucleotides decide who we are and what we are. So imagine there is a thread, and you are connecting some 4 different color of beads. The number of beads, the arrangement of beads, the order of beads will decide the organism and the nature of the organism. So those functions are carried out by all those 4 nucleotides. And those informations are not exactly identical between 2 organisms or between 2 human beings. So if I take myself and himself, we both have 3 billion base pairs, and we have 99% similarity, meaning these 4 nucleotides will be arranged in the exact same order for myself too and himself too. But the remaining 0.5%, we have difference. Like that may be, if he has cytosine, I may have thymine. That constitutes our differences. He is a male and I am a female, and he executes his function differently and I execute myself differently. Okay. So these variations are called genetic variations, and it is very common between individuals and between organisms. So analyzing this genetic information is the key to study how a particular organism behave, or how we can manipulate the gene. So I said there will be genetic variation. The nucleotides will change between person to person or between organisms, and there are many different kind of variations. One is substitution, where one nucleotide will be replaced by the other, and sometimes the nucleotides may be deleted, or sometimes new nucleotide may be inserted. Sometimes they will be flipped. So all these kind of variations we can see in the genome of any organism. Yeah. So, like, is it boring or okay? Okay. Okay. So this variation, sometimes if it is in the critical gene region, it may cause disease. For example, if you see the hemoglobin beta gene, there is only one nucleotide change where the amino acid glutamic acid is changed to valine. So that makes our red blood cells to become a sickle cell shape. So this can cause a disease. Even a single change in the nucleotide can cause a disease, or such changes. Here is another gene called ApoB 3A and 3B. These 2 genes are present in all the human beings, but in some of us, the entire ApoB 3B gets deleted. So among this group, 1 or 2 people may not have this ApoB 3B. That doesn't affect the function of our cell or the physiology of our cell, but it may associated with increasing the risk of cancer. So the third one is, there are other changes in the genes which are called polymorphisms or genetic variation. They didn't impact us much, but they can give us information. So who our parents are, or where we belong to, whether I'm an Asian or an African, those kind of information I can get from these genetic variations. So now, up to this point, in a nutshell, every organism has a set of genetic information. We call it genome. And within the genome, most of the sequences are similar. We also have some genetic variation. And those genetic variations, meaning the nucleotide changes, it may cause a disease, or it may associate with the disease, or it just simply present there, and it can tell us some information about ourselves. So these three are the major things about genetic variation. And genomics is actually studying all those things. So at first point, we collect the complete set of genetic information, and then we study the variation, and then we interpret based on our need, whether we are analyzing a disease or whether we are checking with the ancestry. So all those things we can get from this genetic variation information. So as I mentioned, this variance will help us to identify the genetic diseases, and it can also help us to identify disease susceptibility. Imagine a population of human being. We are all same, but we are very sure, right? Our immune system or our immune response is not the same. So some are easily affected with cold. Some will never get cold in their lifetime. And even when there is a viral pandemic like the COVID-2, some will get affected and recovered faster. Some will be succumb to the viral infection. Those things can be influenced by the genetic variant the person has. And then this genomic variance will help us to predict our ancestor, who we are, like even from just one generation. I can find out my parents. At the same time, I can find out my very long back ancestor. Even if I check my genetic information, it may be possible I may share a huge portion of Denisovan DNA. So from today to some million years before, we can get all those information just by analyzing this genomic variance. So now, I hope you get some clear picture. We have a complete information and there is a variation, and that variation is our focus because we can check whatever analysis we want from those variants. And how we are doing it. For example, I am a geneticist. I can ask you to spit on my face. You can do that. And I can scrap your saliva, and I can get the complete set of information, your genetic information from that spit. So your spit, the blood, your hair sample, everything has the blueprint of genetic information of one person or one organism or whatever. So once I collect here for decency, I have shown blood. We have collected blood and we have isolated DNA from them, and we do next generation sequencing. Sequencing is like, as I mentioned before, the same example, you have a thread and you have 4 beads of different colors. You were given a reference, right? I am giving an 1 meter long thread and a specific order of those 4 beads in that thread. I gave it to you, and I have some other thread and broken pieces of beads. And I ask you to arrange exactly the same that I gave you. So you can use my thread as a reference, and you can arrange the beads one by one. So this is what happened during next generation sequencing. So there will be a reference sequence. If I am analyzing a Homo sapien, there will be a reference sequence. From there, you can arrange the nucleotides of the sample you have. It may be anything, blood of a modern human or blood of a Neanderthal, or you can have some ant or bug or any DNA. You can just arrange it with the reference sequence, and your complete genetic information is ready. So imagine if it is a 3 billion base pair. It's very difficult to arrange in one go, right? So usually we break them into pieces, and then we arrange them, and we have a tag called ligand, and we connect it. That would be make the job easier, right? So this is how the complete set of genetic information is taken from the sample. So how this technical advancement was done? In the year 1990, in US Parliament, there was a discussion like we should sequence the human genome to see how advanced we are. So the scientist panel decided to sequence the human genome. At that time, the facilities are very meager. That's not what we have today. So they think it may take some 15 years or 20 years to complete the sequence of a single human genome. And then they have an announcement, we need volunteers to give the sample for our sequencing. But luckily, their plan was to complete the sequence, the very first human genome sequence in the year 2005. But before that, in 2003, they assembled everything. And after that, the sequencing and genomics field grew very faster. Within 1 year or two, they started sequencing all the organisms they see, like from Drosophila, the housefly, to mouse, to C. elegans, to whatever they see, they sequence their genome and arrange it. So this is the key information. And now the technique becomes so advanced. Before, if you take sample from an individual, to sequence their complete genome, it may take 2 weeks or 5 weeks or so. Sometimes it may take months because our computer facility is not so advanced. But now it will take just 2 or 3 days. So the genomic technique is so advanced. So we can sequence in just 2 days. Now, all over the world, there are millions and millions of human samples were sequenced for genetic information. And it's very mandatory that all those information should not be kept in privacy. It should be available to the public. So we have plenty of databases with human genome information. Can anyone guess why it should be made public?
The research.
Yes, mainly for the research purpose. And if someone wants to discover some or identify some drug or some new disease, then they can easily take the information and then they can execute their research. If it is kept private, we don't know, right? Every time we have to sequence the same person again and again, so which is very tedious. So it's very mandatory that today, if I sequence someone's genome, I have to enter into any of the database. Either it belongs to Indian database or it belongs to the international one. That sequence should be in the public. That's the major thing. So these are some of the databases. Even you can, it's not clear, right? So one is UCSC Genome Browser, and this one is International Genome Consortium. So you can just browse it in Google and you can see the genetic variations that Americans have, Africans have, and the Indian population have. So now we know the importance of genomics and we have the technique of genomics, how to sequence samples. Even like we have a very small fragment of DNA. When we see the Denisova cave, all the fragments of DNA were almost degraded. In those conditions also, we can retrieve them, we can get the complete genome. So we are advanced to that level. Now we can see how we are going to use it. The first primary aim of human race is to like beat any disease, like Maranamilla Peruvalvu. That like everyone wish for that, right? So our primary aim is to like we have to live and we have to live happily. We have to live without any disease. So whenever there is a disease, we will try hard to beat it. So even the field genomics is also used to identify genetic diseases. Okay. Before I have mentioned about the sickle cell anemia. That one we mentioned as single gene disorder. So if there is a single variant in only one gene, hemoglobin beta gene, there will be a disease. So, and the symptoms are very obvious. So that person will have anemia because their red blood cells are not functional. For a doctor, it is easy to analyze and it is easy to diagnose and set the patient for particular treatment because the cause is very simple. But there are millions of human beings around the world. They have some symptoms. Doctors are not sure about it, and they don't know the reason. Throughout their life, they will move from one hospital to the another and to the another and spend millions and millions of money to not for the cure, just to know why they have such condition. So we don't know that. We say they are rare genetic diseases, which is very not common, which is not in any medical books. And those kind of genetic diseases remain undiagnosed all over the world. So if a family has it, they have to bear with it. When the genomics field arrives, there is a solution to it. Because, for example, I think you may not heard about the disease citrulline anemia. No, right? Okay. Citrulline is one of the metabolite in our system. Whenever citrulline is deficient, there will be anemia. Like deficiency of citrulline is citrulline anemia. And it happens in the liver. And can you guess, the symptom appears in the neurons. So they behave like they are mentally abnormal. But the only thing is, instead of citrulline, their bodies start accumulating ammonia. So if you consider a developed country, they know what citrulline anemia is and what the symptoms they show. So immediately the doctors over there can suspect this particular patient may have citrulline anemia, and we can give treatment according to that. The treatment is very simple. They should stop eating ammonia producing foods like peanut and some other foods. So they should stop that. That's it. It's almost a dietary control. But many underdeveloped or developing countries, we never heard of those disease. And what doctors could see is their mental abnormality. So immediately they will ask them to meet some psychiatrist or psychologist or get admitted in some mental hospitals. So those kind of patients will never come out of it. They don't know what they are affected with. In such cases, before, the diagnosis is very rare and expensive, and we have no clue how to diagnose. Now, with the genomic information, the doctor can ask, I can't diagnose with the previous knowledge, so you can get your genome sequenced. So with the genomic information, you will get all the thing. Maybe this person is deficient for citrulline, so he or she may be affected with citrulline anemia. Then the diagnosis may be better and easier for the patient. This is one such example. There are so many things where like people have diseases which doesn't have any name also. So as I mentioned, they are very rare. It may be like 1 in 1 lakh people or 1 in 2 lakh people or even less than that. And many people are suffering by it, but they don't have proper diagnosis and they don't have proper disease management. So this is an example how genomics help to diagnose one such rare genetic disease. This one was executed in our lab. I think there is a rare genetic disease called Cowden disease. It affects 1 in 200,000 people. So you can guess how rare it is. And if the person has that disease, he or she may develop cancer in multiple organs, from the gastric portion, from the skin, from the oral, and everywhere. Throughout their life, they have to encounter the formation of tumor, and they have to go for cancer therapy. There is no other go. And the previous diagnosis is look for a gene called P10. P10, we say, is a tumor suppressor. So whenever one of our cells try to become a tumor, this P10 gene will be functional and make it to stay calm. So it will not allow our normal cell to enter into a cancerous stage. So it's kind of a guardian of our genome. The diagnosis is such a way, if you see multiple cancer in a person, screen them for PTEN mutation. If they have PTEN, then we have to go for other option. If they don't have PTEN, then obviously it is the reason. But I did my postdoc in Japan. In Japan, there is a family where the father, this one is father, and they have 3 daughters and the 2 daughters. Father and the 2 daughters were affected with Cowden disease. But when the doctor checked for their PTEN, it is fully functional. So now we can't diagnose the reason for why they have Cowden disease. And there is another doubt, should it be a Cowden or some other disease? We don't know that. So we plan to sequence the complete genome of all the 5 people using next generation sequencing. And we found out that their PTEN is fully functional. They are a good behaving guy. But there is another gene called AMRA1, which is nowhere related to Cowden, that showed up. That may be the reason this family has the symptoms of Cowden. And then we proved AMRA1 may escalate the Cowden symptoms using cell line and zebrafish. And if you want to read about it, you can use this paper. And now I can register AMRA1 gene in the database. So if someone in other part of the world have the same symptom, then they can guess if it is not PTEN, it may be AMRA1 or it may be another gene from some other part of the universe. So this is one example how a particular gene can be narrowed down in an arid situation. So after this one, we now know there are many genetic diseases which went undiagnosed and also which went untreated. Before I mentioned, we have so many databases to store our genetic information. And after the rise of awareness of rare genetic diseases, again, every country, even the small one or the big one, have multiple rare genetic disease databases. They have the complete list of genetic diseases they have, and they have the complete list of gene information. So if one of the patient in a very small village have some symptom, we can directly go to the database and we can check to suspect a possible disease. So when the genomics become rising, the whole universe is networked. All our genetic information can be shared. All our disease information can be shared. For example, imagine if there is a thief in a street. If we live very isolated, we don't know, right? Oh, and the வீட்ல 5 நாளைக்கு முன்னாடி வந்தாங்களாம். But if we are so networked, அந்த 5வது வீட்ல ஒருத்தன் குதிக்கிறான் பாரு அப்படி சொல்லிட்டு அப்பவே அவனை அடிச்சு துரத்தி விட்டுருவாங்க. So genomics is also similar to that. We are so networked and all our informations are available for public. So whenever we have a difficult situation, we can get help from anywhere, from US or from Africa or from if we are in Chennai, we can get information from Virudhunagar or anywhere. So it's only possible because of the advancement of genomics. And then the second one, we have seen we can diagnose very rare diseases using this field. So what is the fulfillment in it? So if a patient come, உங்களுக்கு இந்த disease இருக்குப்பா போய் படுத்துக்கோப்பா அப்படி சொன்னா it's not enough, right? We should have a treatment option. So what is next? Can we use the same genome for treating the disease? So for that, we have a new technique called CRISPR-Cas9. And have you heard of this terminology somewhere? Okay, yeah. They received the Nobel Prize for this CRISPR-Cas9 thing. So, for example, if in a genome there is a small portion which has a variation, and this variation is causing a disease. So now, with the use of CRISPR is some sequence, some DNA sequence, like the arrangement of 4 nucleotides. And Cas9 is an enzyme that has the ability to cut the DNA. Okay. With this CRISPR-Cas9, I can just cut this portion of DNA and insert a new portion with a normal one. So now the mutation can be removed from a person or from an organism. And also at the same time, if I need a new behavior in some organism, for example, I have a mango tree, it is always giving very sour fruit. I want to improve it, then I can edit its genome using CRISPR-Cas9 to make it produce more sweeter and more pest-resistant mangoes. So CRISPR-Cas9 is again a very vast area. So here we just see how it is used for treating a disease. And we exploit, like we are Homo sapiens, we are very smart animals. Whatever we see, we can exploit for our survival. So we don't leave a bacteria or virus or some other neighbor species. If it is advantageous to us, we will exploit it. If it is dangerous to us, we will kill it. So here, the CRISPR-Cas9 is taken from the bacterial immune system. You know, whenever we are infected with some virus or bacteria, there is a well-developed immune system. It will fight, right? It will kill the bacteria and it will kill the virus to protect ourselves. Bacteria has high chance of infected with viruses called bacteriophages. So the single bacteria will develop immunity so that the same virus cannot attack it again. You know, if we see how it happens, when the virus enters into a bacterial cell, it will not enter as a whole. It will just pass on its genetic information. So think of this is the bacterial cell wall, and a virus is infecting it and introducing its DNA into the bacterial cell. And now the bacterial immune system, say CRISPR-Cas9, they will cut the viral DNA into pieces. So by this way, the virus cannot survive. And then think about the intelligence of bacteria. It will take a small portion of the viral DNA called spacer and attach it with its own DNA. Can you understand, right? So first it degrades the viral DNA. The job is not over yet. It will just take a portion of viral DNA and have it as a reservoir. Can you guess why? Yes. Yes. In the future, if the same virus infect, it already has the information. So it doesn't need to proceed from step 1. Oh, it can attack easily. Now, how we, like human beings are smarter than bacteria. So how we are exploiting it? We know spacer DNA. So, and this spacer DNA is very important for the Cas9 enzyme. Cas9 enzyme is like a scissor, it will cut DNA fragment. So if a Cas9 enzyme sees a spacer, it will cut. So if we make, for example, if there is a gene, and if a Cas9 sees a spacer here, it will cut. So we will make our own genome as so many spaces. For example, if I want to edit hemoglobin gene at position 2300, okay, so then I will make the position of 2200 as a spacer for Cas9 enzyme. So I will introduce Cas9 so that this enzyme will cut my own DNA, and then I will supply another DNA fragment with the correct nucleotide that will be inserted by our DNA repairing machinery. So this is how CRISPR-Cas9 works. So simply, we are exploiting the bacterial intelligence. So as I mentioned before, this one is a spacer and this one is complementary, is similar to the target gene. For example, maybe someone is affected with beta thalassemia or sickle cell anemia. So the hemoglobin beta gene will be targeted. This sequence will be the same as the hemoglobin beta gene. So once it makes binding, then the Cas9 enzyme will cut this portion into two. Now I can introduce the DNA fragment with a correct nucleotide. This can be inserted and the error is repaired. This is how CRISPR-Cas9 is used for genome editing. And now officially this gene therapy is approved for treating sickle cell anemia in USA. And now India is giving some more time for more research. Maybe in future, we will have gene therapy for sickle cell anemia and beta thalassemia. So this is the initial point of using genome editing for treating a genetic disease. So in future, we will have a lot of gene therapies for complex diseases also. Before, I always mentioned about single gene disorder. If you think about cancer, it has multiple defective genes. So those kind of diseases also we can get treated with genomic editing. The research is going on, but it may take some years to happen. So, and now we have seen like there is a disease, we can diagnose it, we can treat it with the genomic information, and now we can, it's almost 1 hour. Okay, I will finish it faster. Okay. 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 We can also use the genomic field for genetic susceptibility. It is not so boring, right? Okay. Okay. Like, can you guess what is disease susceptibility? Yeah.
The likelihood of a person getting this.
Yes. Like we can say this particular person may or may not get the disease. For example, this is a very common saying. அவன் ஒரு நாளைக்கு 10 சிகரெட் குடிக்கிறான்ப்பா, அவன் நல்லா இருக்கான், அங்க பாரு அவருக்கு எந்த ஹேபிட்டுமே இல்லை, அவருக்கு கேன்சர் வந்துடுச்சான் அப்படின்னு சொல்லி, திஸ் இஸ் வெரி காமன், ரைட்? திஸ் கைண்ட் ஆஃப் திங்ஸ் கம்ஸ் அண்டர் சசெப்டிபிலிட்டி. அந்த சிகரெட்டே பிடிக்காத நல்ல பர்சனுக்கு, சிகரெட் பிடிக்கலன்னா நல்ல பர்சன் சொல்ல மாட்டேன், பட் ஹியர் ஃபார் ரெஃபரன்ஸ், அந்த பர்சனுக்கு ஹிஸ் genetic makeup will make him susceptible to get cancer. So we cannot blame his habits. We cannot blame someone else's habits for his disease. So, okay. There are many variants, right? Some variants will make us too susceptible to a disease. Here, this one is a previous example where I mentioned this particular gene is completely deleted in some people. Maybe like with the disclaimer, we cannot share our genetic information in public. So it's like a kind of privacy thing. I should not share my genetic information openly with you. But here I can say, I have in my genome, in everyone's genome, there will be 2 copies of genes. Okay, if it is apobic 3A, there will be 2 copies of apobic 3A, and it's 3B, there will be 2 copies. Whatever gene you take, there will be 2 copies. And I said this deletion is a bit common, and some may have 2 copies deleted. In my genome, I don't have 1 copy of apobic 3B. So there is no like physiological impact of this deletion. It is very common in a population, but see, you can see the black one represents the deletion. So if you see the Africa and the Eurasia, deletion is like very rare. But if you see the migration of human being from this to this, the deletion is more predominant. And based on the population, this deletion in some population, it is very like neutral. It will not affect the person. But in some population, it is strongly associated with cancer. For example, if you take Southeast Iranian, Chinese and Malaysian, Indian, this deletion is associated with breast cancer. The person with this deletion may have higher chance of getting breast cancer. But if you see other cancers in the same population, there is no association. So it doesn't have so much influence over it. So when we study the genetic variation, we can also find out whether we are susceptible to certain disease or not. And this one is another interesting thing regarding the susceptibility. And maybe during the COVID period, this paper was all around the news. No one remember that? Okay. We know, right, during the second wave, Delta, European countries were much more affected. At that time, genomic studies of both the virus as well as the human being were at peak. So there are two reasons. One, to study the viral genome, we want to know how faster it gets mutated and it gets more virulent. And to study the genomic information of host, that the human being, the important reason is which population is more susceptible to the viral infection, which population is not. And this is a very small study, but a very good one. They just checked 3,200 individuals and they found that there is a region in chromosome 3. See this one. Say, if you see this is the length of chromosome 3, here a portion of genome, if that portion is present, if the variation in that portion is present in a particular person, then he has a higher susceptibility to get more severe COVID and to get hospitalized. And another interesting thing is this portion came from Neanderthal DNA. So it doesn't belong to the Homo sapien. Maybe some million years ago, during the mating of Homo sapien and Neanderthal, this genomic variant gets passed to the modern human being. And now in the year 2020, that DNA makes us susceptible to COVID infection. So now I think you can admire the field of genomics from present day to past, and then you can come back, you can shuttle with the information, right? So this is a very interesting discovery. And one of the author is Babo, and he received a Nobel Prize for analyzing the Neanderthal. Actually, he discovered the Denisovan fossils, and he is the pioneer of paleogenomics, and he developed the protocol for analyzing the archaeological samples. Yeah. I mentioned about this portion in chromosome 3, which makes us susceptible to COVID infection. Here is the prevalence of it. You can see wherever the infection is very higher, there we can see like the frequency of that chromosome 3 Neanderthal portion. And then like I mentioned, the very serious one, we have to diagnose diseases and we have to treat it. And the second one is like, am I susceptible to cancer or am I susceptible to infection? Everything can be done with genomics. And again, this is kind of fun part. We can detect your own ancestry using this genomic information. There are some commercial bodies available where, as I mentioned before, you can just spit your saliva in a tube and you can send them. They will analyze your ancestry and let you know, oh, you belong to Africa, maybe some 10 generations before, your forefathers are from Africa. Or you can even discover like your unknown ancestry. You may be a Filipino or you may be a Brazilian. All this kind of information you can get by, but you can pay and get to know your genetic information. And in India, 23andMe is somewhat common, but India doesn't have this kind of commercial thing so far. We are more focusing on disease diagnosis and treatment. But if you want to like sequence your complete genome, you can still do it, and it may cost $1,000 US. So you can have all this information, but I recommend not to do it, because it may say like உங்களுக்கு gastric cancer susceptibility இருக்கு. Susceptibility doesn't mean you will definitely get cancer, right? Then you have a psychological thing. You will always think about, ஐயோ இன்னைக்கு உளுந்த வடை சாப்பிட்றோம், gastric cancer வந்துரும்டா அப்படி. So like let it be where it is. And yeah, this is what I wanted to mention. So you can screen, anyone can screen their entire genome if that particular person has a family history of genetic disease. If they have breast cancer, the mother has breast cancer or the sister has breast cancer, then I can go for my genome to be sequenced to be on the safer side. And if you want to know your curiosity, ஐயோ மதுரையில தான் இருக்கும்னு நினச்சிட்டு இருந்தோம் போல இருக்கு, நம்ம ஆப்பிரிக்கன் போலவே அப்படின்ற மாதிரி இன்ஃபர்மேஷன் you want to know, then you can go for checking for fun. And like yeah, many in US will do, like they want to know what their genome is and they will just sequence their genome. And other thing I want to mention here, you know, like many people think all our behavior and all the thing we do comes from our DNA, right? So even அவன் புத்தி தெரியும் அதெல்லாம் டிஎன்ஏல இருக்கு அப்படின்னு, but it's not like that. If a particular character is influenced by a single gene, for example, if you take eye color, it is influenced by a single gene, then it will definitely have strong DNA influence. கருப்பா ஐ கலர் இருக்கிறதுக்கும் ப்ளூவா இருக்கிறதுக்கும் there may a single gene is responsible for it. But if you consider intelligence, that's not because of a single gene. We have 23 pairs of chromosome and each chromosome has a lot of genes which code for our intelligence. நீங்க பாத்தீங்கன்னா தெரியும், there will be many genetic diseases like down syndrome ஆகட்டும், Turner syndrome, whatever genetic disease a particular kid has, their mental stability will be bothered, right? Mental abnormality is a very common symptom of any genetic diseases, which means all the 23 pair of chromosomes have some genes which code for intelligence. And I always say, if a particular character is controlled by one gene, we can disturb that gene or to change that character. If a particular character is influenced by more than 10 genes, then another player comes in, which is called environment. So for example, like there is a very intelligent kid, his or her genetic makeup is so good, her father is a Nobel Prize winner or whatever, but if you don't bring him to school or if you keep him inside a room for years, there is doubt that he may develop education or he may develop intelligence. So stopping him from studying or not giving or not providing the proper environment will not make that particular kid intelligence, right? So whatever character is controlled by many genes, environment plays a crucial factor. So now what happens is, maybe the bad side of genome, we all try to get the complete genomic information, and there is another group, they put race in it. See, genome of white person is so privileged. It's like error proof. It codes for the most intelligent genes. So that's not at all correct. That's absolutely insane. All the intelligence and your behavior is controlled by multiple genes, and environment plays a major role in it. So those kind of interpretation we should not make from your genomic information. So far we have seen like the application of genomics and how far we have traveled in this field. Now we see what is happening in India. What are the steps taken for the Indian population by our government? The one is, once the Human Genome Project was completed in 2003, scientists from all over the world, they were very curious to sequence all the human beings so that the complete information is known. So first they developed a project called 1000 Genome Project, meaning DNA will be collected from nearly 1000 people and sequenced so that it will be considered as a sample DNA. So if we have any suspect for disease, we can go to that 1000 genome and get information. So every country has their own 1000 genome project. India also has it, and it completed its 1000 genome project in the year 2020. So if we go to the Indie Genome database, we can get all those variations specific to the Indian population. And the rapid development, immediately after 2024, we have our 10,000 genome sequenced. So we improved the sample size and we have a lot more information of our Indian genome, and we collected 10,000 samples, and all over the nation, 20 institutes are playing a major role in it. Now we have our own Genome India, where we have nearly 10,000 genomic information. And if you want to see, like just play around the variations, you can go to this website, cleangen.igib.res.in, or just enter 10,000 Genome Project India. You can, in Google, you can just go to the website directly. And for rare genetic diseases, India is having a national policy for treatment. If you are so curious, if you are a curious researcher to diagnose or to treat rare genetic disease, you can write to this policy your research proposal. They'll immediately accept it, because identifying, diagnosing, and treating rare genetic disease is a key all over the world. India is also heading to it. And there are organizations for rare diseases in India, and they have complete information. Say, for example, some people have some disease, they put their information in it. So on the other side of India, if I have suspicion, I may have some rare disease, then I can go to this organization and get help from them for proper diagnosis. And this one is from two scientists, Sridhar Sivasubbu and Vinod Skaria. They themselves created this project to have all the genetic information of rare genetic diseases that are frequent in India. And as I mentioned before, for genome editing, India is ahead for treating sickle cell anemia using CRISPR. Maybe in 5 years or so, we also have gene therapy to treat sickle cell anemia and beta thalassemia. And that one is like some 34 crore project aiming to be completed in 5 years. So far, we say like we can get our genomic information for $1,000, or we can see the disease, we can cure the disease. And what are the legal things behind it? Any researcher or any person, if they come to you to get your genomic information, say, I want your blood sample, or I want your saliva sample to find your genomic content, then it is very compulsory that they should provide you proper informed consent. Why are they collecting your sample, and what are they using it for? So if I say I am a genetics researcher, I collect blood sample from one person saying I am analyzing the cervical cancer risk in that patient. Then in a due course, I am using it for another study without informing this patient, then it is like illegal. So that person can sue me. So the informed consent should clearly say everything about it. Maybe in the future, I can use for some other disease. Every information should be given to the volunteer who is ready to give their own genomic information. And yes, of course, it should be always made public. Then only we can do some drug discovery, or we can do some disease identification. And there is always a debate whether, say, for example, if I have 10 genome sequences, should I have a patent for it or not? So that's a very long debate happening all over the world. And we have seen we can use genome editing using CRISPR-Cas9 for treating disease. But using the same technique for human embryo has ethical concern. So far, it is ethically not approved. have you heard of the China case some 3-4 years before? So all over the world, the DNA is considered so natural and we should not disturb it. That was the primary thing of a geneticist. I can study it, I can edit in a cell line, or if there is a very severe disease, I can edit for a particular person. But the very important thing is it should not affect his next generation. The DNA correction that I made in one person, it should retain within that person. It should not move to his son or daughter. So that one was a universal rule. But in the year 2020 or so, I guess, maybe even before that, in China, there is a researcher. He edited the human embryo for some good thing. So the mother has HIV infection. So he edited the genome of the embryos, I think twins, so that the kids doesn't have AIDS. So that was a good thing, right? But it should not be done in human embryo. For example, imagine anyone can misuse this technique. Right? They have like superhuman being, so they can edit their own embryo. So we should not entertain all those things. So that Chinese researcher was immediately arrested. Actually, his paper was published in Nature for his like very bravo thing. But immediately he got arrested, and he served his sentence in jail for 3 years, and he came out recently. So editing human embryos or doing genome editing that can pass to the next generation is completely ethically not acceptable. And apart from that, genomics has so many role in animal breeding, plant breeding, and metagenomics. Like you can swap your gut and you can analyze all the bacteria and viruses in one go by sequencing everything and then separating them. Oh, this belongs to E. coli, this belongs to some other virus. So you can do everything in one go. That field is called metagenomics. And these are all helpful for preserving biodiversity, or if there is an endangered animal, we can sequence their genome and we can store their information. In future, if possible, we can revive back the animal using some other technique. So I'm not going to talk all this thing. So in a nutshell, we can use as a normal person, we can use this genomics for genetic testing to check whether I have a genetic disease. Some people have a family history, so we can easily go for genomic testing to make sure I don't have the disease. And for ancestry prediction, and for like even some people use lifestyle modifications. Now there is another field called nutrigenomics. So they will sequence your genome and they will recommend the food you have to take. So if you wish, you can go for it. That is not at all a bothering thing. But we cannot use genomics for the wrong side. One is all will come under the category called screening. It is not diagnosis. It is not done by a doctor saying like, unga genome edutittu, ஆமாப்பா உனக்கு இந்த டிசீஸ்லாம் வரும் போப்பா. You should not do that. You may have this disease. It doesn't mean you will definitely have it. As I mentioned before, environment will also play a factor. So you cannot take the genomic result directly as diagnostic result. It is just a screening. And you should not use, one should not use that for racial discrimination. ஏய் அந்த ஜீன்ல எனக்கு இருக்கிற டிஎன்ஏ பத்தி உனக்கு தெரியுமா, அதெல்லாம் உன் ஜாதியில இருக்கா, அப்படிலாம் பேசக்கூடாது. So that both doesn't have any connection. And another thing, we should not connect it with pseudoscience. The above is from from a religious guru. He said he knows all information about genomics and he can do some puja to genetically distance yourself from your forefathers. How can we do that? How can we do that? So just by knowing scientific terminologies, we cannot cook some new information and we cannot use it as a pseudoscience peddling. That is strictly not acceptable. When I saw that tweet, I was like, இப்படி எப்படிப்பா கனெக்ட் பண்ண முடியுது? How can you distance yourself from your ancestor? இது எப்படி possible? And another pseudoscience peddler, he said he is a DNA astrologer. He said இப்போ வந்து கிரகம் அதெல்லாம் தூரம், உங்க உடம்புக்குள்ளே நீங்க என்ன ராசின்றதெல்லாம் இருக்கு. I can predict and I can say. So, this is impossible, right? So this kind of pseudoscience things should be avoided and should be considered with care. உனக்கு ஜெனட்டிக்ஸ் பத்தி என்ன தெரியும் அண்ட் எப்படி அது ஒரு ஜீன் ஃபங்க்ஷன் பண்ணுதுன்னு தெரியாம யூ ஷுட் ஜஸ்ட் யூஸ் த டெர்மினாலஜி ஃபார் யுவர் ஓன் ப்ராஃபிட். Thank you. You can ask me any question you want.
Thank you so much for giving such a holistic outline of what these gene, genome, and genomics are about, and also for talking about the ethical concerns that are that we deal with in this study. So we'll open the floor for questions. We'll take a few questions and then we will answer all of them together.
Thank you. Thank you.
Yeah.
Nice lecture. I'm just talking, I want to ask a question regarding gene therapy.
Okay.
You said you have gene therapy available for sickle cell anemia and beta thalassemia. What about cancer genetics? Is there any gene therapy available for cancer? Specifically, I'm talking for breast cancer and cervical cancer. Because we have identified the gene.
Yes.
BRCA1 and BRCA2.
Yes.
But at the moment, what we do is if there is available, if we do detect that gene, we provide clinical guidance to the patients. But is there any gene therapy available for that?
It's gene therapy is not available, but okay. So I have to collect all the questions, right?
Few questions.
Okay.
Sorry?
Oh,
What is this?
okay. So therapy is not available, but it is research is being taken place for all type of cancers. Like, because even in breast cancer, if we see BRCA1 and BRCA2 are the major genes, there are some other genes also playing a role. So they are the major drivers of cancer, but behind that, there will be many passenger genes that influence the breast cancer. So gene therapy to it is very difficult, but people are trying to do it, editing multiple genes at once. So that's what cancer research is going on now. Maybe it may take some years for gene therapy for cancer.
So you've identified the deletion of the BRCA gene.
Yes.
Like you said over there, example.
Yes.
Take out a bit and
Yes, we can.
trying on that, or that's just.
It's like very early stage. They are doing it in some animal, are doing it in cell line, but no clinical trial or no no relevance with the patient.
I have a question with regard to CRISPR and how you can modify genes to modify your characteristics and so on. Are there applications in, say, making designer babies or making superhuman beings for defense purposes or offensive purposes like the army and so on?
Yes, we can. Like, if we take technically, we can do it, but ethically, we should not do it.
Is it legal as of now?
No. We should not touch the human embryo at any cost. It's not approved.
Hello, ma'am. My question is, if I sequence and study this cell right now, and if I sequence the same cell after 2 or 3 years, can I figure out the accumulation of the mutation in the cell?
Yes,
Like
yes.
If, for example, if I sequence the cell and I found the gene variant as a heterozygous, and then after 2 to 3 years, if I sequence the same cell, can I find it to be pure or homozygous?
Yes, it depends on what cell you are using. If you are using a cancer cell line, any cancer cell has the nature to change its genome so often. So if I sequence the cell today and I see some variants are heterozygous, meaning only one copy is changed, in a few years, I can see both the copies are changed. That's very common in cancer cells. But if you take some other cell, like red blood cells or white blood cells, at some point of time, red blood cell will shed its nucleus. It doesn't have any DNA. And if some cells which are very resistant to cancer, which doesn't multiply or which doesn't proliferate, in those cells, the variation will not occur. If it is heterozygous, it will always be heterozygous. It depends what kind of cell you are studying.
So in normal cell, it is not possible.
Yes, it's not possible unless you are introducing some mutagenic agent. You are exposing the cell to UV or you are exposing the cell to some viruses, then there may be a possibility. But if you are just growing the cell, it will always be heterozygous.
Thank you.
Yeah.
Good afternoon, ma'am. It was nice hearing you.
Yeah.
Do you think consanguineous marriages, that is marriages that happen between blood relations, could lead to some rare genetic diseases?
Of course. Of course, yes. You know, like consanguinity, meaning marrying the relative, will always increases the chance of getting any genetic disease. And even if it is rare, it increases the chance of getting it. For example, we have a patient, they have warfarin syndrome, which is very rare actually, which is very common in Indonesian islands. But in Tamil Nadu, people are having it. A particular family is having it because of their consanguinity. So it will make the chance to be increased. Thank you.
Ma'am, still the traces of Neanderthal target us. It's not a racial basis. Like I doubt whether we are COVID scenario, still the traces target naturally. It's happening. And one more question is whether there is Denisovans. Those traces are here. We found in Hillary or somewhere like.
Okay.
Just
Yeah. The Tamil people may still have the traces of Neanderthal DNA or Denisovan DNA, but we don't know it because we didn't sequence our genome. And when we think about Hillary, so far we have extracted samples of like 4,500 years old. So that is very recent compared to the whole picture of evolution. 4,500 is just like a 4-year-old kid. So the genetic information that we get from Keeladi will be more similar to us, the modern human, than to any other archaic human, meaning our cousins like Neanderthal or Denisovan. Some people who are interested in Keeladi research, they are like very curious, like Neanderthal or Denisovan, which are unique species of human being found in some places. Our Tamil land may also have some subspecies, right? We didn't know it. Maybe we have to dig deeper to get samples of some lakh years before, then we can confirm whether we have our own subspecies or not. But as of now, the genetic information is not so like elaborate.
Hello, ma'am. When a child is born, it has like Down syndrome. If it's born with Down syndrome, we've commonly heard that it's because of the mother's probably like becoming stressed, or it's because of her anxiety or something like that. That's what I've commonly heard. Is that true? What's your take on that?
Obviously not. So for the Down syndrome, so if we say syndrome, it related to chromosomes. One chromosome may be completely absent, or one chromosome may be completely present, or part of chromosome may be absent or present. So the reasons for it is very late marriage. It belongs to both father and mother, right? if the age is so like above 35 or above 40, the chance of getting a Down syndrome baby is high, but it doesn't related to the mother's stress. Even for autism, like people say, it is because her mother is always stressful. So whatever happened to the child, it is because of mother stress. It's not like that. There are multiple reasons, and so it is not.
Hello.
Yes.
You were mentioning the importance of making a genetic database public and its uses in the future. But is there a chance for misuse of this particular information, as in the application of biowar in designing genetic weapons, which could potentially be lethal to the whole population? And how exactly is the legal consent for this particular process before making the data public?
Okay. So first I'll mention about the legal process. Say it is public, but a common man cannot use every information, right? First, they have to know about it. And if you enter into any database, the basic informations will be available to any people. So if you go deeper and deeper and deeper, you need some designation. Either you should be a researcher, or you should be a scientist or a professor, and you should hold some account to open the deeper information about some samples. So it's not like directly available to all the public. And for the biowar thing, say if that's like technically very complicated, right? If I have a population of 1 lakh people and 100 may have some disease susceptibility, then I have to design a disease to make them infected or make them affected. And even then, the remaining people are safe, and we have some treatment options. So making use of genomic information for biowar is like not so easy. We can just directly spread the virus or directly spread the bacteria to the whole population. Anyhow, some will be dead or some will be infected. So that's easier. Oh, sorry, I'm not giving idea for a biowar, right?
Hello, ma'am.
Yeah.
Is there a gene compatibility test for partners prior to going marriage? I thought, I mean, I've heard somewhere, but is that like a correct thing, or is it medically proven?
It's not medically proven. Of course, people do bizarre things. Like once they get some like information, they will just play with it. Of course, there are people who do gene compatibility with their partner. It is not medically proven, or it is not genetically okay, because you cannot match 3 billion base pairs, right? It's just for fun. Yeah. Maybe some people use their blood group for checking the compatibility. That's all, like just for fun. It's not scientifically so serious.
Ma'am, you said yeah, the Denisovans and the Neanderthals DNA were sequenced in the case. So is it possible to revive them back?
Maybe we are there, like we already have their residues, right? That's enough, I guess so. We cannot revive them completely, because like maybe in Jurassic Park movie, just with the genetic information, we can bring back the dinosaur, but it's a very complicated thing. Like we have to develop the cell, we have to develop the organ and the whole human being, and then the brain. So that's technically impossible. So maybe if there is some nerd scientist doing it, then it's okay. It may be possible.
A continuation to
Yeah,
this
yeah.
question, Mark. Is it as over generations the genetic information is dispersed over a large population, collecting specific genes that orient to specific characteristics of the
Like
cavemen,
okay.
the cavemen, is it possible to construct a complete genetic sequence and then replicate that into a modern human? That wouldn't be ethical, but then is it possible?
Yeah. Like you mean to say, like, we have their genetic information and then we are like,
Yes, collect pieces of genetic information and then form the whole transcript. And I haven't watched the movie, but yeah, maybe. Yeah.
okay, you are reversing the evolution. Okay, yeah, we can do it. You don't need to do it genetically. You can just stop them being educated. You can just make stop them eat cooked food, or and they will. So I was like, it's just the environment, right? Even though you have the complete transcript of a Neanderthal and you are producing a Neanderthal man here and you are giving him education, he will be a modern human being. So it's just the environment. You can just change it. So all your questions, I give some weird answers. Sorry.
So I'm okay to disclose this. So in my in fifth chromosome, I have a one band inversion. So I've been treated for it. That's not a problem. The question here is, in the scope of genetic editing, when you say editing, does that technically mean the inversion can be undone, or you completely take that out and then replace it with the actual nucleotide in the proper order?
Okay.
So whether you turn it upside
No,
down, is
like,
that possible?
We will just take that portion out and put
Complete-
the normal portion in. That one is easier.
Okay.
Yeah.
Thank you.
Well, of course, we are very curious to go back and wonder how things were and if we can reproduce the past. My question is slightly forward-looking. In the coming years, what can we expect in medicine, the developments in medicine using this field of study?
Okay. So theoretically, most of the single gene diseases can be cured with gene therapy, but so far it's super expensive. So a normal person or common people cannot meet it out. Maybe the billionaires, if they have the disease, they can get cured. But in due course of time, it will be available to everyone. So this will be in one track. The treatment for single gene disease will be made available to the common people by policies and government. At the parallelly, as Sir asked, the gene therapy for complex diseases like cancer or which involves multiple genes will be taken care of, and research will be so in a progressive way to meet it out. It depends on the disease. Mostly there are multiple genes. Yeah.
So ma'am, you said that like basic characteristics such as an eye color might just be coded by like one gene. But then when it comes down to like personality, behavior and all of that, multiple genes interacting with each other do affect it. But it's so normally said that because the parent is like that, the child is of the same, like has the same talent or so. Maybe more extroverted, more introverted. How much is the environment and the upbringing only affecting that? Is there a play of genes in it? Like, is it encoded inside them as well?
Okay. So for this question, I'll go with technical answer. There are two terminologies in genetics. One is heredity and the second one is heritability. So both are different. Heredity means whatever the information, genetic information my father and mother have, I will have it. So that is heredity. So it comes with like eye color and all those things. Even some level of thinking ability I can get from the parents. But heritability is like mostly that particular character is influenced by the environment, but there are some genetic variations, just one or two nucleotides in the father and daughter or father and son. It may influence their behavior. For example, like Ilayaraja plays music so well, so do Yuvan Shankar Raja. So இங்க வந்து பாரு அப்பாவும் நல்லா மியூசிக் போடுறாரு, பையனும் போடுறாரு. So it may be genetic. It is not heredity. இது வந்து அப்பா கிட்ட இருந்து பையனுக்கு டைரக்டா வரல. It is the environment. Yuvan Shankar Raja, by the day he bonds with the music. So, and also there is a chance he has the heritability. 1 or 2 genes which are responsible, which may be responsible for music knowledge, they have small variations that both Ilayaraja and Yuvan Shankar Raja may have, that which I don't have. So that makes difference. Like appamo smart, payanu smart, or mother smart and daughter smart, that may be heritable, but not inherited exactly. Like, am I clear? Okay, okay.
It's usually 50-50 or generally?
No, it's not 50-50. Yeah. So even I have like
Given the environment is an art.
yes.
In case it's 80, it's much more.
Yes,
that
yes.
we can-
If in many families you can see smoking father or drinking father. If that environment is so horrible, then the son or daughter will not smoke or will not drink, because he realizes the environment. It is very bad for the family, so he doesn't do it. But in some cases, even though he lives in a very bad environment, he may think, அவங்களும் குடிக்கிறாங்க நானும் பண்ணலாம். So it may push an urge to drink. That may be heritable, but it is not like genetically very sure. அப்பா குடிக்கிறதால தான் பையன் குடிக்கிறாங்கன்னு இல்லை. It is mainly decided by the environment, but here and there are some reasons, genetic reasons.
Okay. We'll take maybe 1 or 2 questions and then we'll wind up and you can approach the speaker and then ask. So.
Thank you so much for the talk, ma'am. I just had one small question.
Yeah.
I've heard in the news that because of the parents' use of a certain recreational drug and because of their addiction, the children are also born addicted to the drug, ma'am. How does that happen relating to genomics?
If you ask me, I would say it's mainly because of the environment. Like the child sees like they are using the drug, or they are like exposed to the drug at the very early stage and they start using it. But very strong genetic association is not there.
Thank you so much. So the Anna Centenary Library would like to honor our speaker today. So if you want to get information regarding the Tamil Nadu Science Forum Popular Science Lectures, please give your information to us, or you can approach any of us for any other information. And thank you so much, ma'am. So if you have anything that you want to discuss, please approach our speaker. Thank you.
Again, thank you for
Thank
you for the opportunity, and thank you for being so interactive. Yeah.
Okay. So, ma'am, you asked 2 times whether the session is boring or not. I think there are a number of questions, I would say. It was a wonderful session, ma'am.
Thank you.
Actually, so நான் தமிழ்ல எங்க தாத்தா ஒன்று சொல்லுவாங்க, தாத்தா பாட்டி கூட்டம் பூட்டி ஓட்டிக்கு அப்புறம் வரவே இல்லைன்னு சொல்லுவாங்க. This session, it shows that I have a very long relationship between my brothers.
Like
So,
we don't know, we all have own house now, but definitely our ancestors have own cave. We have our own property.
Yeah, it was very much informative actually. I believe students and those who are very much interested in this topic have a lot of things to learn more, and so much information is provided to them. And if you have any questions, you can approach her personally, ask your questions. And apart from that, Ma'am Revathi Devi is our EC member, TNS of EC member. And I just learned that she's just finished her maternity. She has a 5-month-old baby, and they are down actually, waiting for her. But she wanted to take this session. It shows her dedication. Yeah, that's what I want to highlight here. And thank you so much, ma'am, for your presentation today.
Thank you.
Yeah, thank you all for being present here. Thank you.
Mm.